Hello everybody, this is for B-Day, September 29. I'm out for district training and won't be here today. And I'm recording this video to help you understand what you're doing for the lab. So the first thing you want to do is you want to locate your data table. I taped them on the tables so there's no mix-up. There you go. Once you're done with yours, please take it off and you can throw it away. Next, please open your computer and go to Canvas and click on the lab, the Potato Core lab. You want to download the PDF file and then open it with Kami. I'm going to pause right now to give you time to do that. And once everybody has that file on Kami and you're ready to roll, you click on Continue with the video. You will scroll down to get to the data table 1.4. Please note that this table starts with distilled water on the top and then goes to 1 molar sucrose on the bottom. Your table may set up the exact opposite the other way, so please make sure that you get the numbers correct for the initial mass and the final mass. In grams, you don't have to write grams here, just the numbers. So for example, if the potato that was in the distilled water had an initial mass of, let's say, 5 grams, and then a final mass of 6.5 grams, this is how you write it in there. You can pause the video now, and I give you time to write that all into the table. And now you start your calculation. So get a calculator, and this is actually pretty easy, and we're going to go row by row, not column by column. That will take you longer. We have to find out the difference between the masses, and to find that, we type in the final mass, which in this case is 6.5 grams, minus the initial mass, which is 5 grams, and then you hit enter. And the number that you get, you write that down where it says difference in mass. Don't delete that from your calculator. Take that number that is still in your calculator, so that was the difference, and divide that by initial mass. So in this case, that was 5 grams. Make that number times 100, hit enter, and that is your percent difference. Please round it to a whole number and write that into the percent change. Do not write any percent signs into the cells. Pause the video now and fill out this table. You don't have to put any class average in right now because you don't have that. But you want to get the percent change for all of your potato cores that you had in your lab. Next, scroll down to table 1.5. This is where we're going to collect all of the data from every group. You can black out the row that says team members. We don't need that. And we also do not have 8 groups, so you can black out group 8 as well. Once you have all of the 6% changes, the first 2 should be positive and the last 4 should be negative. Send one person from your group to the board and fill in your information right here. Once all the information is on the board, please copy those numbers into your own table 1.5 with Kami. Calculate the class average. So you're going to add up all of these numbers from all 7 groups. Here goes the total. And then you divide it by 7 because we had 7 groups, and the class average goes in here. Again, round it up to a whole number. Pause the video here until all of this is done. Now that we have all the class averages calculated for the 6 values of the concentrations, it is time to work on the graph. So go to the next page, and there's a graph. There's a piece of graph paper for you. In the middle, you will see the x-axis. So the value 0 here, that's where y is 0. And the x-axis is for the concentration, the molar concentration of the sugar water. It goes from 0 to 1, so 1 is our biggest number, and the 1 goes on the last line, so right here, and every 10 lines is 0.2, okay? So between here and here is 10 lines that are going vertical. Every 10 lines, 0.2, another 10 lines, 0.4, another 10 lines, 0.6, another 10 lines, 0.8, and then another 10 lines, and you're at the 1.0 molarity, and at the last line, that's on the x-axis. For the y-axis, please use every 4 lines 10, okay? So 4 of the little lines will make it 10. Well, if you go down, of course, that's negative 10. If you go up, that's positive 10. So 1, 2, 3, 4 lines is 10. And you probably only need 20 or 30 on the positive side, and on the negative side, it will go down to 50. So pause the video here and establish the scale on both x and y axis. Now that we have the graph paper ready, we are ready to plot the data. This is what you do next. You put dots on the graph paper where your data is. So for example, at 0 concentration, that might be somewhere here. And at 0.2 concentration, this might be somewhere here. 0.4 could be here. 0.6, 0.8, and 1.0. When you look at the dots, you will already see that this will not make a straight line. We do not want to connect dot to dot because then we get a curved line. What we want to do is we want to make a line of best fit. A line of best fit has approximately the same number of dots underneath the line as you have on top of the line. So I made a line here, and you see a couple of the dots are kind of underneath it and some are on top of the line, and that will work as a line of best fit. Pause the video until you have done that. Okay, you have your line of best fit. Now let's talk about what's happening here. So you know osmosis was happening with the potato core. When the potato core was in water, that in distilled water or 0.2 molarity, you saw that it gained mass. That means water went into the potato core and we had a positive % change. These are those 2 data dots here. And then after a while, when we used a higher concentration, we had a negative change in the mass and therefore a negative % change. And the dots are below the x-axis. So when we look at above, we know that when the potato cores gained mass, that means water went in. So osmosis went from the beaker into the cell. But if we look at these numbers down here, we have the opposite side, the opposite movement. So water went from the potato core into the beaker, and the potato core lost water. Now, what do we call that when the potato core and the water that it's in, or the solution it's in, has the same concentration and there's no net movement of water going in or out? Pause here, discuss it with your friends what that's called, and then click on play again. If you said isotonic, that would be correct. And where's the cell isotonic to the surrounding solution? When there's no net movement, so that means there is neither a gain nor a loss of water, and there is 0% change. Where this blue line intersects with the x-axis, this is the place where it's isotonic, and we need to find out at what concentration this happened. Did we actually have a beaker with exactly that concentration? No, we did not have that. But because we have that graph and we have some data above and some data below it, we can now tell exactly what that number is. So look at where the blue line, or whatever color your line is, intersects with the x-axis, and determine from the x-axis at what concentration that happened. Everybody's is a little bit different because everybody made their own line of best fit, but there's not a big difference between the numbers. So once you have this number, please write it underneath it on that small line that's where it's asking for that value. This is the line I'm talking about. So it's asking you for the molar concentration of sucrose right here, and that is the concentration that is the same that the potato has on the inside also, where there is no net gain or loss of water. Now scroll down to the next question, question number 1. It's actually explaining to you how we get the solute potential. Solute potential, or Psi-S. If you type it in, you can simply type then SP for solute potential. The formula for solute potential is negative I CRT. Now, what does all this mean? The negative sign has to be there because otherwise we couldn't say that when the solute increases, the water potential decreases, but that's exactly what it does. So we need a negative number for that. And there you have this negative sign. I is the ionization rate, but sugar does not ionize in water. It does not make any ions like salt does. Therefore, the number for I will be 1. The concentration you use is the concentration that you just read off your graph and put on that little line. R is a constant, 0.0831. That is actually on your paper. And T is the temperature, and the temperature has to be in Kelvin. The reason why it has to be in Kelvin is because Celsius as well as Fahrenheit has negative numbers. If we suddenly have a negative number, our whole value will be positive again, and we can't have that. Now go to the box where it's asking you to calculate the solute potential and use this formula. So first you have your negative sign, and then for ionization, since it doesn't ionize, we're going to use 1. We can't use 0 because then our whole number would be 0. So -1 times the concentration. I don't know what concentration you have. That's a little bit different for everybody because of the line of best fit. So put that concentration value in there, where your line comes across the x-axis, where the value for y is 0. That is the value you're looking for. Times 0.0831. That is a constant. You do not have to memorize the constant. Constants are always given to you. And then the temperature in Kelvin. So if we use 20 degrees Celsius as our room temperature, you add 273. That's Kelvin. So 273 plus 20 is 293. So this is what you calculate. -1 times the concentration that you read off, times 0.0831 times 293. Once you have this number calculated, here's your second calculation, because now we're actually wanting to calculate the water potential. And there's an easy breezy formula. Water potential equals pressure potential plus solute potential. Now, did we have any pressure? We did not. That little cup that you had and covered with a lid, there was nothing on here to put pressure on it. And neither was there anything on the potato to put pressure on it. So for pressure potential, we're going to use 0. So then your water potential equals 0 plus the solute potential that you calculated, whatever that number is, and this is a negative number. And if you add a 0 to any number, it's just still that same number. So your water potential is the same number as the solute potential you just calculated, because we're only adding 0. If there was any pressure involved, then that number would change. And pressure would be positive. So if you have, let's say, a negative value of 3 for the solute potential and 5 for the pressure potential, then your water potential would be 2. But pressure is 0, so your water potential is the same number as the solute potential that you just calculated. Next, go to question 1. That's on the next page. Question 1 says, if a potato core is allowed to dehydrate by sitting in the open air, would the water potential of the potato cells decrease or increase, and why? So discuss it with your team members and come up with an answer, and pause the video right now. Here's a hint for this question. What happens when water evaporates from something that's a solution? Because if we're talking about a potato core, the potato core has solutes on the inside, of course. So if water evaporates from the solute, and the water potential means the tendency of water to leave, to go away to some other place, what will happen to the water potential, and what will happen to the concentration of solutes? Next question is, if a plant cell has a lower water potential than its surrounding environment, and if pressure is equal to 0, is the cell hypertonic or hypotonic to its environment? You see how this is a comparison? Okay. Now, will the cell gain water or lose water? Explain your response. Then there's a couple more questions that are really short, and the rest of it is wash, rinse, and repeat. There's another graph, and given us the data already of those are zucchini cells, Again, you do the same thing. You will do your graph, put the scales on the graph, plot your data, make the line of best fit, and again, find where the graph goes to the x-axis, and that is the concentration that you need. You do the same kind of calculation that you just did in the box earlier for that problem. And the rest, I think, you can handle. See you next week.